Recent advances in understanding the roles of whole genome duplication in evolution, written by Carl McIntosh and David A. K. Ferrier. It's an article based on the understanding of Darwin's abominable mystery. Darwin's theory of evolution can explain the slow transition from one type of species to another, for example, apes to humans. However, Darwin's theory doesn't properly explain how you can get such a large variety of vertebrates from invertebrates, or from normal flowering plants evolving into flowering plants over a period of time, showing that it is not just limited to animals, considering these are more unexpected transitions compared to apes to human transitions shown previously. So what made that big jump? In recent years, it was discovered that 150 million years ago, our ancestor had 1433 proteins, which were important in regulating how our analog system worked. 1433 proteins are a family of regulatory molecules expressed in eukaryotic cell types. These proteins have the ability to bind a multitude of functionally diverse signaling proteins.
Okay, so 1433 proteins are dimers, kind of shaped like that. And they're very central to cell signaling because they operate by docking on to pairs of phosphosites on many, many other target proteins. So we had been discovering that, for example, if you stimulate cells with insulin, then what happens is that insulin signaling pathway triggers the phosphorylation of a large set of proteins that then dock onto 1433s. So we've been using phosphoproteomics to characterize all of these interactions.
In order to illustrate what happened, we must start with our ancestor. For the ancestor to be our ancestor, this animal has to have an offspring. Here represents pairs of chromosomes. In humans, most female and male cells hold DNA in 23 pairs of chromosomes. But to make an offspring, each of the pair of chromosomes would need to split to each, leading to the egg and cell sperms, only to have one copy of each chromosome. When the egg and sperm meet, this leads to the creation of an offspring holding one of each chromosome from each parent, therefore doubling the normal amount of DNA. Luckily for us, this offspring survived, leading to a cycle of doubling all over again in a later generation known as paleopolyploidy events, leading to the creation of a new animal that is multifaceted and it has such a potential of having 4 copies of every gene. And so this new animal eventually gave rise to all the backboned animals, such as fish, birds, reptiles, and mammals.
If you have Lego blocks with only have one knob, and you have a whole set of them, you put them together, all you can make is a stick. But if you have Lego with 2 knobs, representing the fact that you've just doubled everything up, and then you put that together, then you can make all sorts of different shapes that are more complex. And if different people have the same set, they'll each make something different.
It took 500 million years to get from multifaceted potential to the great complexity and variety of the backbone animals. Unfortunately, during this, a lot of the quadruple genes had been lost and so led to the chromosomal rearrangement over the millions of millennia, showing that in the present day, human chromosomes are just like mosaic bits and pieces of the original duplicated chromosomes. Overall, this study shows that the huge variety of mammals, reptiles, birds and fish involve in different combinations of the quadruple genes. These proteins being kept or lost in different lineages over the years, showing the complexity of our body, structures and variety of many different types of cells in our body is somehow dependent on us having cells which would operate as a pick and mix network from different sequences of proteins from families which are contrasts from each other.